Okay, we've got our exam on Wednesday. A few things to note. So I'm just going to cover up through what I'm going to talk about today about mutation and gene repair. So just like first 10 minutes or so or lecture, the horizontal gene transfer, or what the chapter your book calls how asexual prokaryotes achieve diversity. That section, I am going to talk about it today, but it will not be included on the exam on Wednesday. I'll draw the line. I don't want to talk too much today about what I'm going to test you on Wednesday. So 11.7a homework, that's just a couple of questions. I talked about Griffith's transformation experiment, as well as that experiment, the Avery McLeod and McCarty experiment. That we did talk about. That was the beginning of our genetics where we started. The 11.7 is the horizontal gene transfer, and that won't be due till next time. So that's a little bit confusing. So just wanted to draw your attention there. The dates are right in your homework. The exam's going to have 62 questions. So the last exam was only 46 questions. A handful of people finished in like 20 minutes. I know some people took the whole time. More questions, that still works out to about 45 seconds per question. And some of the questions are very straightforward definition type questions. So you should be able to do in like 15 or 20 seconds. I counted, there's only like 6 questions that I would consider really wordy. So I try to kind of take that into account. I think this is pretty doable without getting all nervous and feeling like you have to rush. But I will have a countdown on the clock just so you can kind of like, okay, am I about halfway done? And about halfway through class, that kind of thing. But so we'll have to be a little bit more cognizant of the time. I had so many things that I wanted to ask you about. So that's why. And more questions there are, the more you can get wrong and still do well. So, okay. I wanted to also revisit this slide because I had a few questions that students asked over the weekend. Number one, a lot of you are asking me about, do we have to, how are you counting the numbers here of ATP and this and that? I don't want you to get too hung up on the numbers. I'm not likely to ask a question, well, how many this or that? The main idea is if you look at the Krebs cycle here, oh, we're getting lots and lots of electrons carried by the NADH and the FADH, and those get changed into lots of ATP. And I wrote this wrong down here. Nobody said anything about this. What I meant to say was 1 NAD is going to convert into about 3 ATP at the electron transport system, and 1 FAD would convert to about 2 ATP. Okay, so I got that mixed up. Nobody said anything. So, okay. Further questions about that? Anything? Okay. So sorry, I didn't recognize that right away. Oops. Okay. So I think this is where we left off last time. Okay, so we had talked about kind of finished up translation and regulation, operons, and then a little bit, just a short section here on mutation. This is one way that prokaryotes can introduce some diversity. It's usually going to be bad. Usually they're not going to survive, but maybe once in a great while that would make a cell more adapted to live in some kind of environment. So we looked at those different kinds of missense, nonsense, silent mutations. You should know those definitions. And thinking a little bit about what kinds of things cause mutation. We think of radiation, and we also think of different chemicals. So looking at the two different kinds of radiation, there's ionizing radiation and non-ionizing radiation. And what's the difference there? I'll come back to that slide in a minute. But the shorter the wavelength you get, the more energy. So remember, wavelength, one crest to another crest. As those get closer and closer together, higher energy. And then whether we call that ionizing radiation or non-ionizing radiation, it depends on the kind of damage that's done in the cell, actually. So those aren't like hard and fast wavelength lengths that correspond to non-ionizing, but usually ultraviolet light is considered non-ionizing. And I mean, did I even post this slide? I might not have. Okay. So that's somewhere between 10 nanometers and 300 nanometers considered that's going to cause a certain kind of mutation. And then X-rays and gamma rays, these are ionizing radiations that cause molecules to become ions. It pulls electrons off of molecules. Okay, so that causes an enormous amount of damage and in the cell to a lot of different molecules, but focusing here on what that does to a strand of DNA, it can either break one strand of DNA or even it can break both strands. Okay, so that's a huge problem for the cell, especially when both strands are broken, because then it doesn't have a template to repair that other strand off of. So if it's going to be repaired, it's basically, throwing in, kind of indiscriminately nucleotides and just kind of guessing for, because it doesn't have a template. Okay, so that's usually really bad for the cell. Non-ionizing radiation causes a particular kind of mutation called thymine dimers. And that's where adjacent thymines on the same strand will bond to each other instead of their complementary adenine base. Okay, so why thymines? It's just something unique about the structure of that thymine base that makes that more likely happen. Sometimes it happens with the other pyrimidine, cytosine, but most likely thymines. And you can see if those two thymines are bound to each other, it does cause a little bit of a bulge in the DNA. So if that DNA is going to be replicated, usually the DNA polymerase III is not going to read it correctly. There might be an insertion or deletion. In transcription, RNA polymerase is not going to read that correctly. So lots of introducing mismatches and maybe insertions or deletions introduced if the cell can't repair it. Okay, so we'll talk about repair in a minute. What other things cause mutations? Well, there are a lot of different chemicals that can cause mutations. I want to draw your attention to a particular kind because we're going to talk about this later in terms of some drugs to treat viral infections. But there's a class of chemicals that we refer to as nucleoside analogs. So remember, a nucleotide is the deoxyribose and the base and the phosphate. A nucleoside is just deoxyribose in the base. Okay, so a chemical that's an analog to a nucleoside, meaning it acts like the nucleoside or is very similar in structure to. Okay, so if something is a nucleoside analog, and it gets incorporated into the DNA. It's not going to, that DNA is not going to be able to be replicated quite the same. Okay, so for example, AZT, azithromycin. You might recognize that. That was one of the first drugs that we came out with to treat AIDS. But it's actually known by a different name now, zidovudine, and I don't know if that's a trade name, but anyway, AZT, this is considered a nucleoside analog. It's got the thymine nucleoside and deoxyribose. It's almost deoxyribose, except instead of that 3' hydroxyl group, instead there's an azide group. Okay, three nitrogens bonded together there. So when this incorporates into DNA, viral DNA polymerases are usually much less discriminate. They would include AZT just like they would a thymidine nucleotide, and it doesn't have that 3' hydroxyl group, so it essentially terminates replication. It can't keep going. So when that gets incorporated, then that's supposed to slow down the replication of the virus. Here's another one, 5' bromouracil, and this is one that they talk about in your book here. We'll draw your attention right there. 5' bromouracil is very similar in structure to thymine. The only difference right here, thymine has a methyl group and 5' bromouracil has a bromine. Okay, this is a little bit confusing here. The numbers, glasses on here, 1, 2, 3, 4, 5, 6. It's just numbering the position there. So this is not have 3 nitrogens and a hydrogen. It's just again numbering the position. So the only difference here is again that methyl group. But if 5' bromouracil is in the presence there when the cell is replicating, it will stick 5' bromouracil in there instead of thymine. And the so what there is that 5' bromouracil binds to guanine as its complementary base. Instead of, remember, thymine should be complementary to adenine. So there's going to be lots of mismatches, the wrong nucleotide coming in there to replicate that strand. And again, eventually enough mutations build up that the cell wouldn't be able to survive. Okay. What else? Oh, here's one other example. Molnupiravir made the news, I think it was right around Christmas time or maybe the first of the year, 2022. It made the news because it was the first drug to treat COVID-19 that could be given orally, that was in pill form. And they actually only narrowly, the FDA only narrowly approved this. It was in the committee, it was like 7-5 was the vote, something like that. Because there were some questions about the safety of this drug, but it is a cytidine nucleoside. So the structure is similar. There's some differences here up at this end and over this end. But basically, again, the idea was to get the COVID-19 to incorporate this. And again, catastrophic mutations, lots of mutations would build up and then it wouldn't be viable. It wouldn't be able to reproduce itself. This was overshadowed. A couple weeks later, another drug was approved, Paxlovid, which has been in much more widespread use because it's much safer. It works a little different way. It doesn't induce mutations. The question came about, do we really want to induce mutations into COVID-19? Okay, obviously, we have had a lot of problems keeping up with the way the virus is changing. Do we want it to keep changing even faster? They said definitely don't give this to pregnant women because it's going to insert into any kind of tissues that are actively growing. So not safe for pregnant women. You wouldn't give this to young kids, but it was a tool that was out there for a while. Don't know whether it really came into much common use or not. Okay, most cells are going to have some kind of repair mechanisms because even though DNA polymerase 3, we talked about how that enzyme has very high fidelity. It proofreads and makes sure it doesn't put in the wrong base. Still mistakes happen, again, hopefully rarely with DNA, at least for our cells. But cells have ways to fix those mutations that do happen. Mismatch repair. This is one that we kind of mentioned before, right after we were talking about replication. And I talked about how methylation typically is going to happen after replication takes place. And we talked about some of the purposes of that. One purpose is for this repair mechanism. I'll come back to that slide in a minute. But so here's the old strand of DNA is in black and the new strand is in red. Sometimes we call that the nascent strand, one that's just been produced. And you can see here in the new strand, there's a mismatch, all right? The old strand was a guanine and a thymine was put in there instead of a cytosine. Okay, so you have these repair mechanisms that go down the strand of DNA and check for mismatches. The question is, how does it know which strand to fix? Does it fix the guanine or does it fix the thymine? Well, the enzyme can sense which strand has been methylated. Okay, and it assumes the methylated strand, again, with that methyl group on it, is the older one. So it's going to fix the strand that is not methylated. So the strand is fixed, that is not methylated. Because that's the newest one. It assumes that the newest one is the wrong one, okay? And then after a while, enzymes will go down, after a few minutes, enzymes go down and will methylate the new strand as well. And then those repair mechanisms are not gonna work anymore. Okay, so that's one repair mechanism. Repair of thymine dimers, most cells have some way to fix thymine dimers because they're exposed to sunlight very commonly. So there's two different methods here. We call one the dark repair and one light repair. We call the one light repair because the enzyme that's involved here, photolyase, is activated by visible light. Okay, so it gets its energy by visible light. So that's only going to happen when there's light present. And basically, we call this method photoreactivation. Basically, this photolyase enzyme goes through and just breaks those bonds of the thymine dimer. The dark repair, again, also known as dark repair, it doesn't have to be in the dark. It can be in the visible light, but those enzymes will work in the dark or light. It's also called nucleotide excision repair. So enzymes will go through and cut the strand where there's that thymine dimer, a few bases before, a few bases after, cut out that whole strand, and then DNA polymerase 1 will come back in and fill in the corresponding bases. Ligase then seals up the ends. So just kind of like remember back with the Okazaki fragments, how it took the DNA polymerase 1 and the ligase to seal up the ends there. Get rid of the primers and seal up the ends. Okay. And the last repair mechanism that I mentioned, I think maybe your book goes into maybe some other ones, but at least in E. coli, they have this error-prone repair that it's other enzymes like DNA polymerase 4 and 5 basically just indiscriminately throw nucleotides onto the strand. That's when you've had like both strands broken. It's kind of like a half-court shot. Like there's this basketball probably isn't going to go in the hoop, but you might as well try. Okay, so in other words, we're probably not going to get the right sequences of nucleotides in here, but it's sort of a last-ditch effort to get some DNA that's going to encode into a workable protein. Okay. All right, so maybe a few offspring survive. All right, so that's where I want to draw the line. Exam, there will be a couple questions about, mutation or repair, but not anything from now on, okay? All right, we have talked a little bit about horizontal gene transfer so far because we've mentioned conjugation a few times, and of course, we talked about Griffith's experiment that showed us that transformation can take place and that there must be some transforming principle. We talked about how we didn't know what that was until Avery, McLeod, and McCarty's experiment that showed that that was not protein, it was actually DNA. Horizontal gene transfer is just one more way that some diversity is kind of worked into the genome. And don't confuse this with vertical gene transfer. So that's when a vertical gene transfer is when a parent cell splits into two and clones its genome into its offspring. Horizontal gene transfer is among cells that are of the same generation. Okay, not necessarily a living cell giving to a living cell, but a cell can be taking up DNA from, again, organisms that are considered in its same generation. Okay, so 3 different kinds of horizontal gene transfer: transformation, transduction, and conjugation. Again, we talked a little bit about transformation before. It talks about it here in the caption. That's where a bacterial cell is going to take up DNA from the environment. And it's usually going to be DNA from a dead cell. Okay, so when a cell breaks down, its cell wall disintegrates a little bit, cell membrane, its contents leak out. And when that is able to, when you have a cell that's able to be, that is able to be transformed, we refer to that as a competent cell. Okay, we think that it's not really all that common in nature for transformation to take place because the acceptor cell has to kind of be able to accept that. Usually the DNA is in a single-stranded form, and then it doesn't get chewed up by nucleases for that new cell. But you think about how that gets transported in the cell, that seems like that wouldn't be an easy process because DNA is pretty long, even in plasmid form. Okay, so we talked about, this is just a little recap, a different textbook showing Griffith's transformation experiment. Remember, that was with 2 different kinds of strep pyogenes, one organism that was able to make a capsule and one that wasn't. And he was able to show that if he killed the cells that made a capsule, if he included live cells that didn't, it could take up the DNA from the cells that could make a capsule and transform the mutants into, again, having them able to make a capsule. So that's just a little recap. We talked about that before. Transduction, I don't want to say a lot about transduction. Know that it involves a virus. Okay, so we're going to start talking about viruses today in a few minutes. We haven't talked about it very much, but there are viruses that infect bacteria cells. So a bacterial cell that had previously infected another host can accidentally package up some of that host's genome and carry it to another host, and that can perhaps get incorporated into the new host's genome. Okay, so this kind of outlines that. We're going to talk more about this replication soon, probably on Friday, so that will make a little bit more sense. Viruses that infect bacteria are called bacteriophage. And not all of them have that weird structure that look like lunar landers, but some of them do. They will usually degrade the host DNA, and then again, some of that can accidentally get packaged up into the new phage that are being made and perhaps carried to another cell and then inserted into that new host cell. Okay, so just for now, know that that involves the virus. And conjugation, we've talked about this because when we talked about that structure, the pilus that some bacteria can make. I think this drawing is a little bit confusing because it kind of looks like that plasmid is being transferred through the pilus. We think that that's not the case, that it's just the pilus is something to draw the two cells together and then a pore opens up. But whatever, we can call it that, or we can draw it that way, I suppose. So bacterial conjugation, what's special about this mode of horizontal gene transfer is that both cells are alive. Okay, the transformation, the cell is taking up DNA from the dead cell. With transduction, the virus is just carrying a piece of a chromosome from the old cell that had lysed. Okay, here both cells are alive, and it involves a cell that is F+, meaning that it has the fertility plasmid, transferring it to a cell that does not have the fertility plasmid. Okay, so I'll write that here. And then that new cell, once it acquires that plasmid, now it has the ability to make a pilus. Okay, the so what there is that F+ cells can transfer other plasmids as well. Maybe like a resistance plasmid. So we think this is one mechanism that bacterial resistance to antibiotics spreads to lots of other cells. I had a question maybe like a week ago, is it always plasmids that are transferred here? I didn't give a very good answer. I said, yeah, I think mostly. Yeah, mostly actually, I didn't say anything wrong, that most of the time it is plasmids, but it turns out, and your book even talks about this, that sometimes that plasmid is actually, that F plasmid actually gets incorporated into the host chromosome. So when that plasmid gets transferred, it can take pieces of the host chromosome with it. And when it does that, it can be known as a high frequency of recombination cell, right? So I wanted to address that just because we had that question, and they do go into that in your book. I'm not too concerned about in terms of what I'm going to test you over, but that, yeah, the better answer is yes, sometimes the chromosome can be transferred as well. And before we were really good at sequencing genomes, that's the way we could map a bacteria's chromosome, by kind of understanding which pieces got transferred and in what order. But I don't think that's done very commonly anymore. So again, now once that plasmid is transferred, now that cell that's receiving is an F+ cell. And these don't have to be cells of the same species. They can be very, very different species, and they can still share DNA that way. So. Okay, let's see, make sure I didn't forget to say anything. Okay, all right, any questions there about, yeah. So what's the purpose of a fertility plasmid? Yeah, I think that it helps it share DNA with other cells. So it's one way that some diversity can be incorporated when the cells are only reproducing asexually. You know, I don't know. I can't really give a better answer than that, but good question. All right, let's move on to a topic that maybe is a little bit more straightforward, and I know for me a little bit more interesting, but we're going to start our chapter today on viruses. Okay, so acellular pathogens, mostly we think about viruses, like they don't have all the structures that a cell, even a bacterial cell that's fairly simple, has. So we're going to start out by looking at kind of the structure of viruses, how they're classified. We're going to think about how there's two different replication cycles that can take place, and then we'll finish. I don't know if we'll get finished next time. We might get finished next time with how to culture them, how to grow them in the lab. And a little bit on prions. Prions are also considered acellular, but they are different than viruses, so we'll be looking at that. All right, so just by way of a little introduction, I don't want to forget to say that I'm going to talk about a lot of specific viruses, but they're just examples. We're interested in the principles right now here. I like to bring in examples because, oh yeah, I've heard of that one, I've heard of that. I'm not going to ask you like specific, oh, what does West Nile virus do? Or what kind of virus is Ebola? Okay, we'll get to that later in the semester. There's a few viral infections that we're going to zero in on and talk about at length. But for now, these are just examples. I think it would be really interesting to sit down and have a conversation like, oh, which of these infections have you had, and tell stories, but probably not really appropriate. But it's interesting we have those kinds of conversations with people we know really well. We think about viruses as being bad, right? You've experienced, some of these, and you know how lousy they make us feel. This graphic is old enough that it doesn't have COVID-19 on it. I don't even think it has Ebola on it, so it's not all encompassing. But there's a lot of different ways that viruses can make us miserable. So that begs the question: why are there viruses? Can they benefit us in any way? Well, one way that comes to mind is that, like I just mentioned a few minutes ago, there are viruses that target bacteria and only bacteria. So there's a potential for us to use viruses to get rid of our bacterial infections. And that has been done, some more so overseas. It's caught on a little bit more than what it is here. We've got to make sure that there's no potential for that virus to do any harm before we do that, use that as a treatment. But other examples, we think that there are some symbiotic relationships perhaps with viruses and other organisms that could possibly be helpful. There's certainly the potential to develop useful biologics using viruses. For instance, viruses have been used as an agent to, or have been used to treat cancer. I know there an article that I read a while back about using poliovirus to try to reduce tumors in the brain. There's potential to treat genetic diseases using viruses as a carrier. And of course, there's been several vaccines for COVID that have used viruses as a way to get that RNA or DNA for the spike protein into our cells effectively. So you have to kind of think for a while, but there are ways that viruses can benefit us. I like to keep this slide because this was the slide that when I taught this class, it was February of 2020, and I said, hey, there's this thing in China that's kind of interesting. Like 500 people have died. Wonder what will happen with that. Of course, at that time, we were calling it the novel coronavirus. Now I updated my statistics here. US cases, 97 million. Of course, now with home tests, I don't know how accurate that number is anymore. But deaths, we're over 1 million deaths now. And that's kind of, man, that just really kind of gets you. So that was the number that I found as of next week. So you all, you know, the age that we live in now, you maybe are tired of talking about viruses, but something that we kind of want to be informed about. Other viruses, Ebola, that one was in the news, what was it, like 2013 and 14, the big, you know, like 8,000 people died in that epidemic. You know, I used to use this slide and say, hey, these coronaviruses, they can cause the common cold. So that's what that one is. Here's a electron micrograph of a bacteriophage, just a virus that infects bacteria. They're just kind of strange looking, a lot of variety in what viruses look like, but they all have in common that they're really small, right? How small are they? Well, if we look at the size of a red blood cell and then compare an E. coli to that, we zoom in a little bit more. The E. coli is way bigger than even the largest virus. Smallpox is one of the largest viruses. And down to poliovirus is one of the smallest ones. So close to the size of a bacterial ribosome. We need an electron microscope to be able to visualize a virus. We didn't come out with those until the 1930s. So we could tell that there was some particle that was really small that was infective by passing suspensions through a membrane with really small pores. But again, we couldn't see them until we had the capability to see them with an electron microscope. All right, so characteristics of viruses. I said they're acellular pathogens. They don't have characteristics that other cells have. They don't have ribosomes, for example. All their cells have ribosomes. They may have a membrane, but they may not. They don't have a cell wall, certainly. No nucleus, no organelles. They are considered obligate intracellular parasites. So that means they have to be inside a living cell for them to reproduce. Okay, so we can't grow viruses up on nutrient agar, right? That is not living cells. We have to have some kind of living cells. They either have DNA or RNA. They don't ever start with both. Okay, so right there, that was pretty mind-blowing when we discovered here's something that can reproduce itself without DNA. In the extracellular state, when it's outside of a host cell, we're going to try to correctly call them virions. Okay, and sometimes that term isn't necessarily used correctly, but we'll try. And then intracellularly, when a virus is inside a cell, it's usually kind of the outside portion of it. The protein portion, for example, comes off. And what you're left with is a nucleic acid. And then we would correctly call it a virus. Okay, so that genome, whether it's DNA or RNA, is always going to be surrounded by a protein capsid. Okay, and we say the capsid is composed of capsomeres. Okay, so you can see that up here in the drawing. Lots of proteins, individual proteins make up the capsid. So we call those capsomeres making up the capsid. Here, for example, DNA, that's the nucleic acid for this particular virus. There may be some spikes, there may not be. I'll talk about that in the next slide. And some viruses are going to be enclosed by a phospholipid membrane. Okay, oftentimes we refer to that as the envelope. Okay, and if they're is no membrane, then we call that either a non-enveloped virus or a naked virus. So again, sometimes they've got an envelope around them, sometimes they don't. A little bit more about that viral envelope. Which cells have them, which cells don't? Commonly, it's common for viruses that infect animals to be enveloped. And by animals, I include humans in that. Okay, so commonly the viruses that infect us do have a membrane. Not all of them, but a lot of them do, more so than ones that infect bacteria. Most of them don't. Okay, where do they get that envelope? Well, they acquire them from the hosts, either from the nuclear membrane or the plasma membrane. Okay, so it's made up of what our nuclear membranes or plasma membranes would be, a phospholipid bilayer. There may be some glycoprotein spikes sticking out from that. So here's the influenza virus. You can see there's several different kinds of spikes sticking out from that. And here's spikes sticking out from a naked virus as well. Those spikes are really important for recognizing certain host tissues, because they're usually really fussy about what kinds of tissues they're able to recognize. So the spikes help them recognize that, attach to their host cells, and maybe even play a role in entering into the host cell. If a virus doesn't have spikes, then those attachment proteins would be on the outside of its capsid somewhere. So it's not absolutely essential to have spikes. And I want to add that typically that envelope degrades quickly in the environment. Okay, so in the environment, I mean on a computer keyboard or on a desktop. Okay. A virus that's, is in the environment like that and it's enveloped, that phospholipid membrane is going to be really fussy, any kind of temperature change, or it's going to dry out really quickly. And without that envelope, that virus is not able to attach to a host cell. Okay, so a virus with an envelope, we typically say it might be viable for a couple hours, maybe up to a day or two. Influenza virus is one that usually survives a little bit longer, maybe up to a day or two. But when they started saying COVID-19 might survive for weeks on your groceries and things like that, I'm like, oh, that's kind of a red flag. That's an envelope virus. Typically, that's not what we see. Something like norovirus, you've heard of norovirus, really nasty throwing up. That is a naked virus, and that can stay viable for weeks on surfaces. So it's a lot tougher in the environment. We had norovirus go through our house a long time ago, and we all got better. And like a week later, my parents came to visit, and then they got it. So it's like, oh man, we needed to clean everything down because that stinking thing just stayed viable for a long time. Okay, so in the environment anyway. What kind of shapes do we see? So we talked about this with bacteria cells, how we see cocci and bacilli and all that. There's three main morphologies of capsids that we see: helical, polyhedral, and complex. And then if those capsids have membranes, then they don't really take on quite that same shape. For instance, here's a helical capsid that's got an envelope around it. Here's some more examples of helical capsids. Remember that capsid, its job is to protect that nucleic acid. Okay, here's tobacco mosaic virus. It's a naked virus, just that long, thin capsid, kind of stick-like shapes. But influenza virus has a helical capsid. It's just got a big envelope around it, so the overall shape is a little bit different. That polyhedral capsid. A polyhedron is a many-sided shape. I couldn't find any examples of polyhedral capsids that were not actually icosahedrons. So that's a shape that has 20 sides. And most often, those 20 sides are going to be equilateral triangles. Remember your geometry, that means that every side is the same length. So that has really high strength and stability. Okay, so that's very popular. It's going to be a very strong structure to, again, keep that nucleic acid protected. And then you're going to have an enveloped polyhedral like herpes virus. Again, just an example. That virus has some other proteins, kind of outside of the capsid and inside of the membrane. I'll talk about that one more later. And then there's this category of morphologies that really doesn't fit into polyhedral or helical. So complex is just kind of a catch-all. It just means that the overall shape is kind of complicated or maybe not symmetric. Smallpox virus is usually considered complex. It's kind of weird structure there. Sometimes we consider bacteriophages complex because they have this tail spindle and tail fibers here. And you can see this head is actually a little bit elongated. These faces here are not equilateral, it's a little bit elongated. Sometimes rabies virus is considered complex because it has this bullet shape. That's actually not due to the capsid, it's actually due to the membrane around it, some proteins in it, but it's not, again, it's not really symmetric. It's not kind of a normal shape. And I just wanted to throw in here, I didn't know where to really mention it, but most of the time viruses are really specific for a certain kind of host cell or tissue, or very specific to only a certain host species. That's not always true, but there's some generalists like West Nile virus. It will infect lots and lots of different species, lots of different birds and mammals. Of course, coronavirus is more of a generalist. You hear about tigers in the zoo getting COVID-19 and pets as well. So lots of different animals there. But a lot of viruses like HIV, if you inhale it, you're not going to get sick. If you eat it, you're not going to get sick. It's only, by certain kinds of tissues in our body that it will be able to infect. But viruses, basically, any kind of animal, there are ones that can infect that species. Like here's plants. This is tobacco mosaic virus. Certain kinds of, only certain species can get that. Here's some bacteriophage. That bacteria cell is in trouble. There's viruses all around the outside of that. And here we've got in the pink, that's human immunodeficiency virus, HIV, that's clamping onto a white blood cell there. And in terms of that genetic material, we're going to revisit this a few times, but it's either going to have, like I mentioned, DNA or RNA. That nucleic acid can either be, it can be linear or it can be circular. If it's linear, it might be in segments, or it might be continuous or unsegmented. So here, this is not labeled, but I think that's an HIV virion. You can see you have 3 pairs of segments there. Coronavirus is unsegmented. It's just one super long piece of RNA. So it's just different ways to characterize that genome. And the genome number is going to be small. In humans, we've got somewhere between 25,000 and 40,000 genes. E. coli, much less. Hepatitis B, that's the virus with the smallest known genome, only 4 genes. So we're going to talk about kind of why that makes sense and what some of those genes are that it's absolutely necessary that it brings with it. And we'll continue that on Friday. So we'll see you Wednesday.